A primary goal of our group is to better understand the connection between neuronal activity with the regulation of energy expenditure and glucose homeostasis. Some recent work has highlighted an important role of both leptin and adiponectin to act within the brain, and specifically within Pomc neurons of the hypothalamus to regulate energy expenditure and glucose metabolism. The results of our current study provide a cellular link to these actions of both leptin and adiponectin within the brain. In the following video, my other co-authors, Jia Sun, Ting Yao, and Yang Gao will further describe the results of our study, and we hope that we provide a better appreciation for the synergistic actions of both leptin and adiponectin to act within the brain to potentially regulate body weight and glucose metabolism.
Recent work suggests that Adiponectin acts, at least in part, in the brain to decrease the body weight and improve glucose metabolism. These activities can be abolished by blocking melanocortin signaling. We found that this is intriguing given the similar roles of the leptin actions in the Pomc neuron to increase the energy expenditures and regulate the glucose levels. Previous work by our groups and others demonstrate that leptin activates Pomc neuron located in arcuate nucleus of hypothalamus. Thus, we hypothesize that Adiponectin may also stimulate a Pomc neuron.
In order to directly test our hypothesis, we generated a report mouse model to identify leptin receptors expressing Pomc neurons. It's allowed for the identification of Pomc neurons which do not express leptin receptors, which were green only, Pomc neurons that express leptin receptors, which were green and red, and leptin receptors expressing neurons, which are not Pomc positive, which were red only. We targeted these neurons for electrophysiological recording, and we found that Adiponectin induced a robust activation of Pomc neurons that express leptin receptors. Conversely, Adiponectin inhibited adjacent NPY AgRP neurons, which occurred at the same time as a decrease of inhibitory synaptic activity onto Pomc neurons.
A recent study suggests that activation of the melanocortin screwed by adiponectin may not only increase energy expenditure, rather this activity may also suppressing feeding behavior. Here our study details a molecular mechanism by which adiponectin can both reduce food intake and increase energy expenditure, while integrating its effects on hypothalamic Pomc and NPY AgRP neurons. To find more about our study, please click the link to the referring article.